There we go. And so I'm gonna now introduce Bill and Jill. Sorry for my doorbell sounds here. But Bill and Jill have actually been working on this project for the past 6 years. I think we were just talking about, maybe even a little bit more at this point, to restore vegetation on Cadillac. And this is something that we, as the park, have been thinking about for a long time, even previous to that. I think Charlie Jacoby had been thinking about this for quite a long time, setting up exclosures up there to keep people off of sensitive remaining patches of vegetation, because we've been noticing trampling. And in those years where we were thinking about it for a long time, we have brought in a number of folks from different places to give advice on how we might approach this. And they all, like without fail, pointed to the odd features of Cadillac, that it's kind of low, relatively low elevation, very thin soils, wind exposed, has lots of visitors, makes it a very difficult place to think about how you would restore. And they all said that they couldn't really give us a say that this approach will work. And they said that we really needed to take an experimental approach. And that's where we got in touch with Bill and Jill and their team with the Native Plant Trust to come in and help lead that experimental approach. And that's what they're going to talk about today, the lessons that we've learned. And so we really appreciate all the work that's been done. And this is a ton of work, so they won't be able to cover it all. But with that, I'll hand it over to you, Bill and Jill. Thank you.
Okay, can you all hear me? Fine. Thank you, Abe. Jill and I are going to tag team this presentation. She's going to advance the slides for me, and she has the controls over there. As Abe said, we started on this project in 2015 with an inventory of the summit, and then for the last 5 years we worked on various restoration experiments. And as for those of you who aren't familiar with Cadillac, it's a subalpine summit at about 1,500 feet, I think, and it was burned over in most of the summit was burned over in 1947. And the subsequent rains washed a lot of the soil away, I suspect. So the soil is mostly either gravel or some sort of light pea gravel, grape nuts, or basically some clay soils up there where it's not bare rock. If you add hundreds of thousands of people walking over the site each year because of its accessibility, then you can see why they might have some of the vegetation disappearing. And so it's mainly fire erosion and people that have caused, I think, the areas up there to be bare. Jill, you want to advance it? So we want to thank, first of all, before we get into this, Native Plant Trust, who contacted Jill and I in order to do this. I was actually working at the Native Plant Trust when the project started, and I've retired since then, although maybe you wouldn't know it. But at any rate, we do want to thank Jesse Wheeler and the vegetation management crew for many, many, many things, helping us with the soil sterilization, moving soil, moving plants. Without them, this project wouldn't really have worked. Abe for facilitating and making sure that we were on track. The staff at Nasami Farm, which is the nursery of the Native Plant Trust, located in the nursery is located in Whately, Mass, for propagation and assistance and expertise, and that covers a lot in terms of taking care of the plants that we were propagating and keeping there at the nursery. Also, Nick Fisicelli, Peter Nelson, the crews from Schoodic for planting assistance. That's been a great help with that, to have all those volunteers. And finally, Friends of Acadia, not last really, Friends of Acadia for helping support, especially with the inventory. Go ahead, Jill. This is a bird's eye view of the summit. You can see it's accessible by car, obviously, and that has been the subject of a number of different regulations over the last year or two. The little black spots you see are all plots we have, and these are areas that are in open rock, open gravel, where we have planted, had various restoration plots, either seed plots or plantings of plants. And what you don't see in this, that all these areas are in fact in exclosures. That some of these exclosures were put up essentially 20 years ago, I think, at this point. But there's not been that much recovery in those areas during that period of time. So basically these are the areas that we chose. We did survey the entire summit for sites around the southwest and southern corridors also, but these are the most open areas and seem to be the areas that get the most disturbance also. Next, Jill. So the goals of the study were first to survey and inventory Cadillac flora. There have been a couple of inventories before. It was very difficult to assess to compare a lot of these inventories in terms of plant species and where they start and where they stop. So Jill will get more into that. To develop and test the experimental restoration methods. Basically, we're using seeds and plants and in various combinations of soils in order to test that. And then finally, to use what we learned here to recommend methods for use on other summits. Jill? Can you advance?
I cannot all of a sudden.
Well,
Hang on.
that's the end of the presentation. Then thank you all for coming.
No, all I did was unmute myself and I can't go up and I can't go down. Okay.
Jill, do you want to, you could either click on your PowerPoint screen. Sometimes if you click on something else, it'll take off the slide advance.
There we go.
There you go.
All right, I can do it that way.
Go back. I think you got to go. Yeah, that's Jill's up next, I think.
Yeah, no, this is, yep, that was you, right?
Yep. That was me. I'm finished.
Yep. Okay. Sorry, friends. Okay. So the inventory was conducted in 2015, and the goals of the inventory were to complete a survey of the summit by collecting plot data along transects, and we actually tried to follow the same transect routes used by Mary Foley in 1992. We collected vegetation data, species presence and cover every, I think it was 5 meters along transects in the 4 cardinal directions from the summit down to the tree line. We wanted to inventory and map rare plants on the summit. The park has always been, in my experience, has been kind of ahead of the curve on doing inventory work in the park, which greatly facilitates planning, because if you know what you have, you can plan how to manage it. So the park already had quite a bit of data, but we wanted to update that. And then in the course of doing the transect work, we wanted to identify areas of intact vegetation. If we're going to revegetate the summit, we wanted to find areas that we thought represented the kinds of associations we wanted to mimic. And so we did that through analysis of the plot data to try to choose which species we would use in the restoration. So the results of that inventory were that we documented 151 species on the summit. We can't exactly do an apples to apples comparison with Foley's data or with Lloyd's data, which happened a few years after that, because a lot of their IDs were only to species. Were only to genus. And so maybe there was overlap, maybe some of the same species occurred there during both surveys, but our IDs were always to species. We were able to update and map the rare taxa records. And maybe particularly important was confirming that Calamagrass districta and expansa, northern reedgrass, which occurs elsewhere in the park, doesn't occur on Cadillac. It was sort of an error of record keeping that happened, I don't know in what year, but it's sort of followed it's been retained over the years, and that species does not occur there, so we don't need to manage it there. The same thing with Mononuria glabra. It's a sandwort, and there's another sandwort that does occur, but recent taxonomic work shows that Mononuria glabra doesn't occur there at all. So I think that's you, Bill.
Yeah. So as far as restoration methods concerned, we used seeds and plants to try to figure out which method was the best for generating plant cover. Now, you can look at this in various different ways. You can look at it from the economy side, which one's cheaper to do it. You can look at it for which one's quicker, which one gives you the better cover. And what we were trying to do was to explore all these avenues to come up with some recommendations for the methods. And as it turns out, you'll see the recommendations are sort of mixed. Next, please. So first was working with seeds. We collected, I should say, Jill and her volunteers collected seeds of the target species from Cadillac. Again, these were the species that we targeted based on the inventory and based on the representation of the species we wanted to recover or to restore on Cadillac. We collected seed, Jill collected seed of 20 species approximately over the years. 11 of these taxa were sown into seed beds. Some of the taxa included black chokeberry, mountain holly, red oak, common juniper, gray birch, spirea, white meadowsweet, euthymia, flat top goldenrod. One prevalent one all over the top of this subalpine summit is 3-2 cinquefoil Subardiopsis trifoliata. And there are various downy goldenrod, bearberry, and again, white flat-topped aster. These are just some of the target species. Those are the 11 target species that we sowed, but there are other species we added, which I'll get to a little bit later. Then we ID'd the sites where the soil loss was documented, and you saw that on that bird's eye view map from previous. Basically, those seeds, those areas were in the north and in the sort of the east of the summit there. And within those, we chose plot locations. And as mentioned, these were also we chose only locations in exclosed areas because we wanted to be sure that we weren't getting visitor traffic through there. Although there were, while we were doing surveys, there were a number of visitors that did show up inside our plot, inside the exclosures, asking what we were doing on occasion. Next, please. So we then took the seed in the fall, we cleaned it, we counted it, we dried it, cleaned it, and counted it at Nasami Farm using NPT staff and volunteers and interns. And we used the germination at Nasami as a benchmark. We then sowed the seed. We sowed the seed outside in plots, usually in either 2 meter, well, always in 2 meter or 1 meter square plots, depending on the year. And we also use various soil techniques, 1 inch, 2 inch, 3 inches of various compost and loam and peat, which I'm sure Jill will get into. But we used they also tested the seed and germination at Nasami. We sowed them into seed flats in the nursery, and the resulting seedlings for that were used to generate plants for the restoration plots. But we also got the germination percentages of all of the same seed that was sown on top of Cadillac through these germination and propagation trials. Now we found that, as expected, field germination percentages is usually lower than what you'll find in a nursery, simply because in the nursery you have much more control over the conditions. But we did find some unusual things happen occasionally. We found that sometimes the seed germination at Nasami, the percentage of seed germination, percentage of seed of a particular species germinating in a seed plot in Nasami was higher than the actual seed germination, seed germination at Cadillac was actually higher than the seed germination at Nasami. And the reason for this had to be either that there was a seed bank in the soil, or that seeds had been dispersed and blown in, or been dispersed in somehow into each of the plots. But it was very interesting. About, I'd say 14% of some of the seed plots had more or suspicious numbers of seed germinating and on Cadillac that made us think that there was additional seed coming either from the soil or from dispersing in. Next, please. So this is just a general germination result thing showing you that the seed germination at Nasami was higher than what a Cadillac. If you look, you can always see that the Nasami average is always higher than the averages of all those same species sown at Cadillac. And you can see the comparisons between the deep and shallow loam and compost. Not much difference between those two when you start averaging out them over time. But basically the Nasami germination, we're using that as a benchmark so that we could compare the germination at Cadillac with the Nasami. And I think that's it for that, Jill.
Okay. So a little background on what we did with those seeds. We, as Bill kind of showed in the aerial, that we had two main areas of plots, but within those areas, there were two what I'll call replicates, but statistically they were really kind of pseudo replicates. But but we had 4 batches of each control, 2 in the site that you saw that was closer to the summit, and 2 batches at the site closer to the North Ridge Trail. So we had a control where we did absolutely nothing except try to demarcate a 2-meter squared area on bare gravel. So we wanted to be able to follow an area where we had done nothing and compare that with areas where we used experimental treatments. We then had a treatment where we put coconut fiber mats, they're called Quire. So we put just a coconut fiber mat over the bare substrate and then held the mat down with these sandbags. We also had one treatment where we sowed those 11 species on the bare gravel, no soil amendment, put the Quire over top, and they're always held down with sandbags. So these first 3 treatments, no soil amendment was added. We also had a treatment where we added bagged Coast of Maine compost. We thought that might be a decent planting medium, and since we were putting soil, we thought moving bagged soil would probably be an efficient way to go. So in our initial 2-meter squared plots, that we had about 1 inch of compost on top of the native substrate. So this one is just compost and no Quire. And then we had a treatment where we sowed seed on top of compost and covered with Quire. So we looked at species richness, so the number of different species, the number of seedlings, and then the percent cover as sort of 3 different perspectives on what happened. After this is I think this one's 5-year average. Yeah, it's 17 to 20, 4-year average. So the greatest number of species were found in the treatments where there were soil amendments. If you look, the difference in actual species counts is like not very much. You know, the control was slightly over 8 species, and these treatments are around 16. But there were more species in plots with added soil, and we all got a big chuckle out of that, because our first and continues to be sort of significant result is plants do better where there's soil. But that result does kind of confirm our assessment of the site. Like, the problem there is that the soil has eroded away. Then we did plant counts, and when we looked at our data, the number of seedlings that we counted in the plots was actually highest in the control, which was moderately upsetting because that tells you that none of the experimental methods had an effect. And if anything, they seem to have had a dampening effect. But then if you look at the number of seedlings and consider only those 11 species that were planted, then we see a treatment effect where the control has the fewest seedlings, and then the treatments that have soil show the largest treatment effect. And the main reason for that is this little plant right here, Hypericum gentianoides, which is orange grass. It tends to come up in that grape nuts gravel stuff like we saw in the control plot. There might be 300 or 400 of these plants per meter squared. They're annuals. They sprout up like 5 o'clock shadow in July. They flower, they seed by mid-August, and then they've all but disappeared. So they're almost hair-like. I doubt that their contribution of sort of organic matter is significant. So I'm just going to go backwards for a second. So you can see how if you adding 300, 600 in a 2-meter control plot would boost your seedling count way up. If you drop out that annual that isn't probably biologically significant in terms of soil building, you can see how the results change. We also looked at average cover. Sorry, my screen is a little obscured for me up top. We also looked at percent cover by treatment. And the blue bars are the planted species only. The orange bars are the all species covers. The plots where we added seed and soil have the greatest amount of cover. And probably there is a treatment effect if you compare it with the rope only control. So I guess that's all I'll say about that. After a year, well, actually, so the first year we put in those 2-meter plots, that Coast and Main compost broke down very quickly, and we were left with gravel kind of strewn with residual mussel shell fragments. So although we added compost, it didn't stick around long. I doubt that it blew away or washed away. We think that it just broke down. So we then tried deeper compost, and we switched down to 1-meter squared plots. So it was just easier to handle. And again, we looked at the same parameters. And we had one area, one treatment where it was still shallow 1-inch compost. And this is versus about 6 inches of compost. And the treatment effects weren't huge. Probably not statistically, we didn't run any statistics, but probably not different than the control. Whoops, sorry. And then the following year, we decided that maybe we should try using loam instead of an all organic soil. We sterilized loam. We did not want to introduce any new species to the summit unintentionally. So the loam was from Mount Desert Island. We mixed it with peat after sterilization. It was sterilized up at park headquarters. After sterilization, we spread compost in seed flats. Bill on each trip would take some of the sterilized loam back to Nasami with him and flat out the loam and make sure that no weed seeds germinated. So that's the story on loam. And the species, there wasn't a big difference in species richness between any of the treatments. That, like, soil depth didn't really have an effect, and whether it was compost or loam, no large effect. In terms of plant counts, again, we looked at all species and planted species only, and the shallow composts had the greatest number of seedlings. Again, that might go back to the seed bank effect that Bill referred to earlier. The one bag of compost was only about an inch deep, and so it may have provided kind of a nitrogen burst, and kind of enabled seeds that were already in the seed bank to germinate. Whoops, sorry. Oh, that's so bad. Sorry, my screen is a little out of control, or I guess I am. And then for percent cover, there was a big difference in percent cover between all of the treatments and the control. Deep compost with seed had the largest number, but again, if we just add soil, that creates a big difference from just the control, and then in this case, adding seed increased it even more. Bill.
So the other type of experiments we did was what we called restoration areas. And this is where we grew plants at Nasami Farm and brought them up there and planted them. These were all into loam peat mixtures in areas devoid of soil. So we put the loam peat mixture on top of the ground, mixed it in a little bit, but basically then planted into about a depth of 6 inches in those particular areas. The plants were watered after planting and roughly for the first month as possible, but really it was fairly irregular. It was based on rainfall. Jesse and his crew did a great job of that, but that was also, we didn't do it for much after a month after that. So I think that may affect the results in the long run. At any rate, I should mention that these plantings were all done in June of the year, and our monitoring was done typically in July and sometimes in September for the plants, for these planted areas for percent cover, to make sure that we had that percent cover for the year. Let's move on. You can see, well, before we move, I'm sorry, Jill, let's go back. You can see if you can.
Yep.
This is basically we started out with areas that looked like they were social trails. We wanted to have areas that we could see the vegetation on either side. We tried to match the plants to the vegetation that was adjacent to them for the most part, and added in other species that were appropriate, we thought, to the particular area as we went. And again, we had to have sandbags to hold the soil in place after we did this. Now, this is an area that was on a particular slope, but many of the areas were much flatter than this when we were trying to do that. I should mention also, we used pots and plugs mainly that we grew at the Sami farm, but we did also try what we called modules, where we put 8 species, 12 in a flat, grew them together in the greenhouse, and then put the module into the planted areas also. We didn't see a great deal of difference between the plugs and the modules in the long run, so we stopped that. But modules might be might be especially useful on some areas that, especially on a slope, in order to hold the plants, and that may hold the soil and the plants together. It may be useful in the near future. Thank you. Jill? As you can see, the percent cover in the restoration plots over time, we were only measuring percent cover. We weren't trying to measure number of plants of each species. Follow that. In the first year, it was a little over 20% when we put them in, and they gradually increased to 47%. The ones we followed, the ones from the earliest plots over the 3 years after they were put in. So went from 22% to 47%, and they increased just about regularly every single year. There were some, if you can give me another slide, there were some areas where we did get some heaving, and particularly with certain plants, vaccinium and rhododendron just seemed out to heave. You can see the pots or the plugs have heaved out and sitting on top of the soil right here. We're not sure exactly why that happens with those two species. Perhaps they didn't form the root balls that the other species did. Although in this final year of the project, we decided we were going to try to do some mulching around these plants just to see if that would help them. We don't have the results of that yet, but we do know that when we did go to mulch, we did tug on all the plants, especially the rhododendrons and the vaccinium, and they were established, rooted into the ground. You couldn't pull them up out of the ground without ripping them up out of the soil. So we're not quite sure exactly why these plants were most susceptible. It seemed to be that in certain areas, and of course, if you think about where you're restoring into open areas, and sometimes these are social trails where you have an idea that there were plants there before, but otherwise, at other areas, you don't exactly know why there are no plants there. It could have been the people trampled there. It could be the conditions of the site mean that plants aren't really going to be able to grow there. And in many cases, we would see rocks, as you see in the background, with a lichen line. So we knew that at least there was soil at one time up to that particular level on the plant. So it gives us a good idea of where plants might be. But on the other hand, there's a lot of heaving. If it's very, very shallow soil and there's rock underneath, you're going to get a lot of heaving as the water sits on the rocks and heaves the plants up. So it just because there's an open area doesn't mean it's necessarily a good spot to try to restore. I think that's the message that we got from that, because you really have to understand what's going on underneath. You can't just add 6 inches of soil on top and expect to get an automatic growth of these plants. Next, please. So the cover and restoration plots versus the seed plots, you can see after the same, measured about 3 years after sowing or 3 years after planting, was much higher, 47% cover in the restoration plots versus 26.48% cover in the seed plots. Now, you know, rain has been variable over that time. There are lots of other factors that could add up into to deciding why these plants and seeds didn't do as well. But in general, you're going to get better cover with the plants to start with, and it's going to increase over time, is what we found in the soil. Next. So when you start looking at this, and by the way, we planted I think 800 something plants in 2018. We planted almost 3,000 plants in 2019, and we planted over 3,500 plants in 2021. We did not plant in 2020 because COVID couldn't find the staff or the wherewithal to get it all together during that July of 2020. So if you're looking at the cost with seeds versus plants, Restoration plantings, we figured roughly cost 12 times, excuse me, more than the same size seeded plots. So you can see you need many more seeds to produce plants than you do in the wild than you do in the nursery. Next. This is especially true when you get to the larger plots, where the amount of seed that you need to sow in a larger area becomes a limiting factor. If you're talking about a large plot like 73 square meters, in a nursery, to produce plants to put into that plot at 6 inches, at 6-inch spacing, and by the way, that's very tight. It's hard to plant at 6-inch spacing. You'll need 6,846 seeds to produce the plants, the 2,062 plants you need. But if you're sowing them on Cadillac, you're going to need 32,000 of those seeds. So you start to find that if you're trying to collect seeds from a local area in order to produce those same size things, that plants, seed is a limiting factor in that. Okay, Jill?
So now we're at the so what stage. Going back to the inventory, our inventory added 50 taxa not listed by Foley or Lloyd. But as I said, many of these taxa were likely present then. It's just that when Foley labeled a grass as Agrostis species, I think we've documented either 3 or 4 different Agrostis species. So we didn't quite have apples to apples, and we don't know how many of those were already there, likely many of them. We documented 15 non-native plant species during the study. The only one that was invasive was one individual of glossy buckthorn that we actually had been working around when Jesse's crew was up there. We had been working next to it all day, and when we kind of sat down and had a drink of water and got ready to pack up the truck, I can't remember who, but somebody looked over and said, "Oh, there's a fruiting buckthorn right here on the very summit. Who knew?" And since then, they've looked from that area and downslope and been treating. But 15 non-native, just sort of weedy but not invasive species were documented in the inventory. As I said, plants grow better with soil. So adding soil to degraded areas increases plant numbers and percent cover. For plots where we added seed, depth of soil was a factor in how good the germination was, but it didn't really translate into increased percent cover after 4 or 5 years. And the planting medium that we used didn't affect cover. Compost and loam had roughly the same amounts of cover by the end of the experiment. After 4 years, species richness was greatest in the soil inquire and soil inquire. So again, where we added soil, there were more different species. As Bill showed earlier, percent cover in the experimental plots increased over time and averaged 26% after 4 years. So, but there was an annual increase. Adding soil with quire results in as much cover as adding soil with quire and seed. So in trying to project forward, what's the best approach to take in future restoration efforts, you could remove the steps of collecting and sowing seed because there really wasn't a difference between adding seed or not. The difference was adding soil. And And then most of the vegetative cover at the end of the study was due to species that weren't intentionally planted. And, you know, we planted those 11 species, but we counted everything. So the bulk of the cover was created by non-planted species. And there certainly was, once the soil was down there in our plots, habitat was created for any local seed rain. Bill, this is you.
So again, average cover at the time of planting was 23 years for plants, increasing to 47 after 3 years. I should mention that the most successful plants, if you look back, the most successful plants that we put in as plants and basically a seed too, Subaldiopsis tridentata, three-leaf cinquefoil, was really one of the most ubiquitous plants and one of the most successful, as was Ulmus viridis subspecies crispa, which possibly does so well because it does fix nitrogen. They either increase their percent cover or remain the same. A number of the other species, including Betula populifolia and Doolingaria umbellata, Euthymia, Graminifolia, Solidago pubilla, and Spirea, had mixed results. Their percent covers increased in some and decreased in others. Surprisingly, Aronia either stayed the same, Aronia melanocarpa, which is up there ubiquitously, stayed the same or decreased over time. And as I mentioned previously, the Vaccinium angustifolia planted and the Rhododendron canadense planted in the plots basically decreased in some cases drastically because they probably because of heaving. And as Jill just mentioned, that it's important to realize that the percent cover in those plots has a lot of species in there that weren't planted and became established and increased the percent cover. Danthonia spicata, we didn't plant any grasses. So Danthonia spicata, Deschampsia flexuosa, and Argrosta scabra were able to disperse and persist in the plots, in the seed plots and in the planted area. And there were a couple of non-native grasses, Festuca filiformis and Agrosta stolonifera, which did also add significantly to the percent cover. So it's not clear, you know, what will happen. It's much more than the plants that we put in there in terms of what's going to happen in the future. So the plant cover in plots where the plugs were planted with loam was always higher than they were planted in native soil. Essentially, planting in native soil really was a disaster. And however, when you're using soil up there, when you're adding soil up there, heaving is a problem. You maybe need to add mulch or coir. We're not sure about that. Next, please. Again, planting plants is much more expensive than collecting and growing seeds, but the results, you get much cover more quickly. And the percent cover of both plants and seeds is increasing. And though for seeds, most of this increase is not from planted species, much more than seeds than, and I think for the planted plants. If you want to compare that to the intact vegetation of the survey, the vegetation of the summit was 62% for those plots that were not impacted by humans or trails or some other factor. So we're at 47% with plants and 20-something% with seeds after 4 to 5 years, 3 to 4 to 5 years, somewhere in there. And we're trying to get to 62, and it's not quite clear how long it's going to take to reach there. Next. Is this me or you? I think it's me, huh?
Yeah, it's you.
So I think we recommend that continued monitoring of this is essential. We realize that there can be small experiments going forward, but it'd be interesting to stand back and watch a little longer before making large efforts up on top of Cadillac. We don't know what the final composition of the revegetation plots is going to be. That'll be a very interesting, in 2 to 3 to 4 more years at least. It will be very interesting to see what the final composition is, and we don't know how long it's going to be to equal the intact vegetation on Cadillac. You may want to consider, if you're restoring up there, you may want to consider seeds in small areas but for large areas and given the limited seed resource, plants seem to be the best and the fastest way to achieve cover. So finally, Cadillac is unique. If you think about Cadillac Mountain, the subalpine summits are probably one of the areas in all of the Northeast, which is under most disturbance from human beings. And Cadillac's combination of unique accessibility, really the auto road getting to the summit, but with the exclusion of visitors in the exclosed areas, and our resources really makes it a unique place in terms of doing this. There's also park staff provided a lot of the help, and the water was nearby. So the whole project really had some unique aspects to it, which would be unique to Cadillac rather than necessarily going to other summits where you wouldn't have accessibility, possibly exclusion, or watering. So Jill, I think you're up.
Yeah, so we took a look, just a cursory look at a few other summits that the park has expressed in an interest in, well, maybe has documented some damage. And, you know, like on Cadillac, this is South Bubble, and you can see clearly eroded on the left. There's just that tuffet of vegetation remaining, surrounded by, you can even see fragments of vegetation around the base of the tuffet where trampling has occurred. And on the right, you can see looks like an eroded bank. So like Cadillac, erosion is a problem on the other summits. The same is true of Penobscot on the left and Sargent on the right. But as Bill said, Cadillac is the only summit with an auto road to the top, and everybody that helped move soil and sandbags and plants worked pretty darn hard to make that happen, just from the pavement to the planting areas. So the logistics of adopting any of the approaches that we identified on these other summits will be more complicated, which maybe makes an argument for monitoring a little bit longer before really committing to a methodology. And so with that, it looks like we have just a few minutes, too few minutes for questions probably, but thank you for giving us the opportunity to do this work and coming today to hear about it. And I can thank you, Kate.
Thank you, Jill and Bill. That was awesome. That was a lot of material that you covered, and I know there was a lot that you even left out. So really super appreciate that, and it represented a tremendous amount of work. So we do have enough time for a few questions. I see Peter has put a couple in the chat. Peter, do you want to unmute and ask one or the other of them?
Sure. Although I would like someone who maybe isn't as familiar with this project to be able to ask a question, but I guess maybe the most interesting one I wanted to point out was that there weren't any non-vascular plants included in your study, which in hindsight, and after spending the amount of time you did up there, do you have any observations about bryophytes or lichens in terms of the regeneration patterns you observed?
Well, there were some plots that had fairly significant, very significant moss cover. To my mind, in those plots, the moss cover looked like it was to the exclusion of vascular plant seedlings. On the other hand, the moss cover probably held a lot of moisture, it did create cover, so prevented soil loss, and might be super important in terms of soil building.
And no, I would agree with Jill on that. And to your other question, Peter, that you had in the chat, wherever it went, there it is. Do you think cuttings from existing vegetation or intentionally damaging the roots and treating with rooting hymns would have produced more colonization? I don't know. Cuttings, I think that essentially you'd have to make sure that the cuttings rooted in order to do that. And it's easier to put plants in when they have roots on them, easier to have them survive than putting cuttings up there. You'd have to put some other, you'd have to think about the wind and and retaining moisture to do cutting. So I don't know about damaging roots with enzymes. I don't know about that process, but I think the cuttings might be problematic from that particular point of view.
And Jesse, do you want to unmute yourself and ask your question? And while Jesse's doing that, if anybody wants to go next, feel free to raise your hand or put a question in the chat.
Yeah, thanks, Bill and Jill. That was great. I guess I just wanted to get your thoughts and recommendations on, with soil being such a key element to having plant cover up their species diversity, everything that you tested. What would be your ideal recommendation for depth of soil, or just how much to get up there to get something established, I guess, and thinking around what plant species were successful as well within that?
Sorry.
You're muted.
You want to talk about that, or do you want me to?
No, go ahead.
I think if you're putting plants in, you really do need the 5 or 6 inches of soil, because the plugs are 3 inches themselves. So you can't go much less than 3 inches where you're planting them under rock. So generally, but so you really need that. But for seeds, I don't know that we have a recommended depth. It might be 3 to 4 inches would be enough. I don't know that for sure, because we didn't really make that comparison. I don't think I'd have to look through the data to make sure if that's really true. I'm not sure about that. Jill,
Yeah, it didn't look like soil depth was that critical. And in fact, in the shallower soils, we tended to have more seedlings, again, maybe seed bank effect. So, as we've said from year one, add soil. And even if it's a little bit, and it has to have some organic matter in it. As Bill said, we didn't show the results, but we did do one year's worth of planting plugs directly in native soil and compared that with our other treatments, and almost every single one of those plants died, and they were watered along with the others. So whatever, it won't do just to take soil, create a borrow pit somewhere, or just borrow soil from other areas of the summit and move it, say, on the bubbles, because I think the organic matter is too low. But maybe some, Peter alluding to lichens and mosses, it may be combining those soil building organisms is something that you'll want to try.
Nick, do you want to go ahead and ask your question?
Yeah, sure. And thank you very much, Jill and Bill, for your presentation and all the work you've been doing up there. And I think both of you, and Jill especially, you've been tramping around all the summits in Acadia and have quite the eye for the different plants growing in these places. So my question was just sort of, was there a species you were surprised to see on the summit on Cadillac doing well, and vice versa? Was there one or more species you expected to see on Cadillac but were not present?
I can't think of any offhand. I mean, based on what the inventory was, Nick, I guess that I think that the presence of some of the non-native species is going to increase up there, and there's certainly, well, I think that the chances of that coming on are, and you're going to see more exotic species coming on up there, I think, is what's going to happen because of the dispersal mechanism from people and cars.
Yeah, can't really think of one that we documented that I was surprised to see or not. Surprised not to see. Yeah, no, sorry. Helen Koch had her hand up.
Yeah, no, I think Helen will go to you, and I think Jesse is maybe going to answer your question, Charlie, in the chat. So these will be our last couple.
Well, I hate to take up time and space with some trivial questions, but I was wondering whether on the Mononuria greenlandica you did a DNA, or were you just doing that for morphology?
Just morphology.
Okay. Okay.
Yeah, with Arthur Haynes worked on that part of the project, and he had been doing a lot of work trying to crack that nut for a while. So he's looked at various populations on the island and feels like even the lowland populations are Greenlandica. But no, we didn't send any tissue for analysis.
Okay.
Yep.
A couple of little other things. After you discovered that this coast of Maine compost was degrading, did you change to a different compost or just add more of the same?
Neither and both. We didn't change to a different compost. We did set up 1 meter plots with where the compost was deeper. So instead of just having 1 inch, it was 6 inches, and that really has seemed to persist for several years. But then, so we did not switch to another brand, but we did switch to our own loam mix.
Okay.
With
Thank you.
loam and sterilized loam with peat added.
Do you mean sterilized or pasteurized?
We call it a soil sterilizer. It's a metal box.
Right. But
It's a metal box with heat.
160 degrees at least
Yeah.
for 3 to 4 hours.
okay, right. Okay, so that's pasteurization. Thank you so much.
Okay.
I appreciate all this lovely talk.
Yeah, thank you guys. So I apologize that we're not going to be able to get to all the questions. So thank you, Jill and Bill, for all the work, for the presentation and the work. And for those of you who are asking, we will be continuing on with the intent of this work was to inform ongoing restoration on Cadillac and the other summits, like Jill got to it there at the end of the talk. And so we'll have more presentations on this and follow-up work for sure. So there will be more opportunities to discuss this. But really appreciate everybody's time coming, and thank you, Bill and Jill, for the presentation, and look forward to continuing to work on this going forward.
Thank you all for coming. Judy, I see you lurking on the sidelines there. Greetings from the East. Thanks everybody for being here.
Thank you all. Appreciate it.
Thank you.